Hello, everyone, and welcome to today's webinar, a game changer for pharmacogenomics, one tool to decode drug response to variation in the genome. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by QIAGEN. To learn more about our sponsor, please visit QIAGEN.com. Now, today's event is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you may utilize this Q&A box, and we will assist you directly. I would now like to welcome our speakers, Dr. Ann Bailey, Lead Clinical Genetics QA Associate Staff Scientist at QIAGEN Digital Insights, and Dr. Elias Hage, Associate Director, Global Product Management, QIAGEN Digital Insights. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Elias, Ann, welcome. It's so great to have you. You may now both begin your presentations.
All right. Thank you, Christy, for that introduction. And thanks, everyone, for joining us today. So just before we dive in, just a brief disclaimer. The QIAGEN Pharmacogenomics Insight, or PGXI, the tool that you're going to be seeing today, is a knowledge-based system designed for integration into broader workflows or systems and is not intended as a standalone product for direct clinical use. And just to give a brief overview, the QIAGEN Digital Insights team has been building for the last 20 years the industry's largest, fastest growing collection of biological and clinical findings. We have more than 200 curators on board that curate scientific literature and have access to public and private databases. So far, we have grown our content knowledge base to more than 26 million unique findings and currently add around 5,000 new variants per month. The content is updated on a weekly basis on our side. And with pharmacogenomics becoming such a hot topic, we are now also expanding our capabilities to curate content for PGX. So pharmacogenomics is the study of how an individual's genetic makeup influences their response to drugs. The genetic variability in certain gene families can affect how drugs are metabolized, absorbed, distributed, and excreted. And this genomic effect can lead to an increase or decrease of drug efficacy and/or adverse reactions. In fact, in a study of over 1,000 subjects, 99% had a genotype that was clinically relevant for at least one medication. And you know that in 55% of prescriptions in the US are influenced by actionable pharmacogenes. Therefore, genomic testing of pharmacogenomic genes can be used to predict individual response to a drug, advise an optimal drug dosing, or even determine if an individual is at risk for the adverse drug reactions. Now, when looking at pharmacogenomics testing, there's two main technologies, with genotypers or microarrays being the gold standard today. But we have seen also uptake of NGS short and long read in PGX testing. Regardless, though, the raw genetic data or the output of those technologies is quite similar. The important piece here is that we look at copy number variants, the variations in specific parts of the genomes, whether they're hets or homs, but also wild-type calls across those pharmacogenes. And for each tested pharmacogene, the genetic result is described using the star allele notation, which identifies pharmacogenomic markers by means of a designated number for a given gene. In fact, many star alleles represent a combination of several variants, and some are defined by the presence of structural variants. And in case of autosomal chromosomes, these take the form of a diplotype by a combination of two specific named alleles. So what you see in this case, an example here, when we look at CYP2C19, which is a gene in this case, we have two different alleles, star 17s, which together form a diplotype. Secondly, we look at, we determine the patient's phenotype, which is really based on the specific alleles present for a given gene. An individual's metabolism or the phenotype is then determined. And looking at CYP2C19, star 17, star 17, this is typically associated with an ultra-rapid metabolizer. And most importantly, is the drug treatment strategy. So the recommendation in pharmacogenomics is typically presented based on the combined phenotype of all tested pharmacogenes, which, if you're looking at CYP2C19, in this case, this would be related to the drug Plavix. Now, pharmacogenomic applications have roles in different segments. In research or biopharma, they can help develop NGS panels, or they can be used in the development of NGS panels that could be tailored to specific populations, diseases, or therapeutic areas. They can be used to stratify patients into subgroups based on their likely response to a drug in a clinical trial. They can also be used for dose optimization in oncology drug development and identification of variations for study design and selection of participants in early phase clinical trials. But they also have a big role in clinical testing, because as individuals age and become susceptible to more disease that require drug therapy, nearly 90% of patients above the age of 70 will eventually be exposed to at least one drug with pharmacogenomic guidance. So PGX testing can be used to predict how an individual will respond to a drug, determine what an appropriate drug dose should be, and if an individual is at risk if prescribed a certain medication or drug. But of course, pharmacogenomic testing has its own challenges. The main ones are identifying the relevant targets and regions, mapping the sequencing results or microarray results to PGX gene haplotype diplotypes, and then, of course, taking those diplotypes and mapping them to drug metabolism phenotypes, and then making a dose recommendation. But to address those challenges, you need access to an up-to-date knowledge base. You want to look at variants from expertly curated genetic datasets and map them to a diplotype, and of course, infer the PGX phenotype and drug dosing recommendation through a platform that integrates and automates that complete process to remove any manual work required. So really implementing a toolkit for pharmacogenomics applications require access to an up-to-date knowledge base, adapting a flexible solution that helps you integrate and automate the complete interpretation process, and of course, something that empowers your decision-making process with access to human curated content. This is where we believe at QIAGEN Digital Insights that the Pharmacogenomic Insights, or PGXI application, will play a major role. It will help you deliver trusted insights with expert verified quality content. It can be used to minimize adverse effects and improve drug efficacy. And of course, the idea is to help to seamlessly scale your PGX workflows with a technology-agnostic toolkit, supporting both inputs from microarrays and NGS technologies. All of this is actually built on comprehensive content to guide the generation of PGX insights. And here I'd like to pause for a few seconds and pass it over to Anne, who's going to take you through a deep dive or an overview of our content and curation strategy.
Thanks, Elias, and thanks also to Christy for the introduction. As with many other precision medicine platforms utilizing genomic sequencing information, the key to accurate interpretation of PGX variants is, of course, the quality of the content that is used. Fully automated content compilation databases can be incomplete or can include lower quality recommendations. Alternatively, there is a myriad of publicly available curated PGX databases, some of which Elias has alluded to already, but they differ in their scope of providing clinically relevant drug dosing information, with few of them being fully comprehensive. On the other hand, PGXI is a comprehensive, expertly curated knowledge base with a standardized process of reviewing and annotating clinically relevant information for PGX genes, drugs, variants, and associating these with the drug dosing recommendation. So what exactly is in the PGXI knowledge base? The PGXI knowledge base leverages over a decade of expertly curated pharmacogenomic content. It supports 18 condition to gene associations, over 300 drug gene associations, around 2,100 PGX gene variant entries, around 19,000 case reference associations, and nearly 43,000 PGX gene genotype associations. The PGXI knowledge base also currently supports drug dosing recommendations for 72 clinically actionable genes. However, the curation pipeline is scalable to expand this curation for additional genes and variants when that need arises. Within this comprehensive content, there are several views of how variants within pharmacogenomic genes are associated with gene diplotypes, otherwise known as star alleles, drug metabolism phenotypes, disease conditions treated by an associated drug, recommendations for altering drug dosage, and the strength of the evidence for this association. PGXI curators utilize a standard curation process that routinely identifies, evaluates, develops, and releases updated content in a quarterly release cycle. To identify new content, curators surveil standard pharmacogenomic reporting databases and guidelines, such as FDA and EMA drug databases, the CPIC and DPWG guideline recommendations, or PharmVar, as well as other publicly available PGX reporting agencies and literature. PGXI surveillance can be initiated by standard monthly or quarterly content update protocols, or through specific customer requests. All newly identified PGX content is evaluated and scored for inclusion into the PGXI knowledge base. This evaluation will determine if there is sufficient evidence of a new PGX gene to drug metabolism association, the strength of the source of the association, as well as the overall strength of evidence for the new content. Once the new content is deemed sufficient to be included into the knowledge base, the new knowledge cases are created for each phenotype to genotype, and new dosing guidance, evidence level, and severity content are annotated. The updated content is released in a quarterly timeframe to ensure that the PGXI content stays relevant and up to date. This curation process creates different knowledge types within the PGXI knowledge base that populates the content for the PGXI data downloads and the PGXI API. Such knowledge types include clinical pharmacology, which provides curated information on drug efficacy or toxicity, genetic variations, which provides information on a genotype to protein metabolism function, phenotype effects, which associates a genotype to phenotype to overall efficacy or toxicity endpoints, and actionability, which defines the overall drug dosing recommendation associated with a PGX genotype to drug metabolism phenotype. And finally, PGXI is earnest in maintaining its quality of its content. In addition to using pharmacogenomic scientists to manage the content curation, PGXI utilizes a standardized quality process for review of all PGX evidence and content surveillance. Furthermore, the PGXI knowledge base undergoes an independent internal review process for newly curated content. Finally, PGXI is enthusiastic to promptly include PGXI customer quality feedback, all of which work together to ensure up-to-date and quality content for PGXI users to use however they see fit. At this time, I'd like to pass it back to Elias, who's going to talk a little bit more about how to access the PGXI.
All right, thanks, Anne. All right, so there's two different ways to really access the content that we've curated for pharmacogenomics. We have what we call a PGX download module and the PGX API. Now, the download module will give you direct access to curated content. The module itself is hosted on our QIAGEN's QDI portal, so customers will be able to have access to a schema, to a bundle of data files that include flat files of all the tables that we have populated, but also a SQL script to help you create that knowledge base locally and integrate it into your workflows or pipelines. Of course, apologies. Users will be able to either download that content directly from a web browser, but we also offer programmatic ways to access that curated content and download it. And the use case to support, what customers can do with the PGX download is to integrate it readily into their internal pipelines or workflows. And then they can run SQL queries to identify what gene or drug associations are available for certain genotypes or phenotypes. We're also happy to announce that there's also a PGX API module that transforms the output of any PGX assays into insights. This PGXI API is very powerful because it's a single API endpoint that is technology agnostic, meaning that it supports input files from arrays, but also NGS technologies in a VCF format, but also other formats from microarrays from Thermo Fisher and the Genome Biosciences. And what it actually does, there's a multi-step process which is completely automated for our users. You'd upload your PGX results, and it's designed to recognize and accept data from any major equipment manufacturer on the market. The files uploaded to the system are then analyzed for their format, and appropriate translation tables are applied to transform vendor-specific data into consistent form for processing. Now, if I divert your attention to steps 2 to 5, that's where I'm going to call it the magic happens with PGXI and our curated content. That's where the data in those consistent formats and genotypes are assigned to help match results to relevant literature. And depending upon the information available for a gene, the genotype may be the zygosity of a single nucleotide polymorphism or a stall allele designation for a diplotype or an activity score. We then actually take the haplotypes and gene diplotypes, map them to phenotypes, and depending upon the gene, the relevant phenotype may describe whether an individual SNP is affected or unaffected, or it may be the metabolic potential of the gene, such as poor or intermediate metabolizer. And step number 4, PGXI API translates the phenotype mapped in step number 3, and with the understanding of the phenotypic variations, the sample data is matched to curated summaries of relevant literature, which is all distilled from the most credible sources available that Anne just showed you earlier. This content, again, includes consensus guidelines from the FDA and other drug labels, as well as summaries of curated research. All that insight that was then generated by PGXI will be made available through a JSON file that could be integrated into your own internal workflows. That JSON format includes insights from the guidelines for drug efficacy, toxicity, and the likelihood of known genetic conditions associated with certain medications. Each insight as well includes a summary of the evidence strength and the severity of issues and reference citations for studies used in developing these insights. And of course, because the output of this PGXI API is a JSON output, customers can then use the internal tools or their own internal reporting systems to customize the output and then report what they deem as relevant and fit in context of the sample analyzed. And just to give you a brief overview of how that looks like, or just some insights on how that could look like on your side. So again, what we provide will be the JSON output, but of course that can be translated into a report on your end. There will be prescribing insights. So for every single medication, what the cause of that alteration would be, what the warning is, the source, and also, of course, references or citations for that curated content. There's also risk management section where certain recommendations will be given, and also what the risk interpretation and the classification would be. And finally, similar to all PGX content out there, or you will be able to get the potentially impacted medications, where you would get informational drugs, ones with warnings, and of course, ones with critical impact. Again, this is just as an example here, but with the JSON output, you and your PGX scientists will be able to tap into this content and then deem what is relevant or fit to be reported from your side. We've also tried, or we are trying to streamline the onboarding process to be as efficient as possible. Because we do believe that the API is the differentiator here, we have built what we call a UI client, where customers during the onboarding process will be able to log into that UI client and then test the API, but also they will be able to support or check for input validations. And what we mean by that, if you have certain files that you wanted to test and see whether they're actually compatible with our API endpoint, you can just load those on that UI client and then troubleshoot with our scientists to identify any issues if that could arise. The API client also allows you to generate API requests and responses to help you also automate that once you are ready to go to production. We also have a services team that, as what Anne was mentioning earlier, will help you expand the curated content in the PGXI KB. For example, if you have any additional genes or variant positions that are currently not supported today in our content or knowledge base, we are more than happy to work with you to expand our curation efforts to include content for these positions. And of course, we can help you and guide you on how to map the JSON output from the API to your own internal reporting systems. And here, just to sum it up all a bit, so the PGX API really accommodates any data from genotyping or sequencing technologies. It will help you support panel design for research or discovery. So with access to the PGXI downloads, you'll be able to query all RKB and find what relevant positions are, or what positions are relevant for certain genes, medications, or even phenotypes, and then use those for panel and NGS panel design as an example. But also will help support labs to efficiently utilize the genetic information and automating the whole PGX reporting process. It simplifies calling of named alleles, the annotation of relevant recommendations, and of course, again, reporting of the findings. And it allows or gives you access to quarterly updated knowledge via either the download mechanism that we just mentioned or through the API. And I just want to divert your attention to the right-hand side of the screen here. We're typically in a manual or semi-automated annotation pipeline. It takes hours for scientists today to go through the whole PGX process. But with the PGX API, or the pharmacogenomics insight product that we're releasing here today, all of those steps in the middle are now automated. So it's as easy as loading a PGX test result. Everything will be automated through the API, and then you're going to get all the content, all the curated content and evidence from our team, which you could integrate and see what relevant information is important to report on your site. So with that, I would like to invite everyone here to go to the link shown here on the screen, and you can request a free trial for PGXI API. We can use, of course, your data, and with one of our field application scientists, we can work with you to see what data is compatible, what coverage or what genes and regions are covered in the PGX API, and of course, give you a sense of how that would look like if you adopt the solution. So with that, I'd like to pause here and open the session for any questions.
Thank you, Elias and Anne, for that informative presentation. As Elias just said, we will now start our live Q&A portion of our webinar. Now, to our audience, if you have any questions you'd like to ask, please do so now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. Okay, so let's get started. And I'd like to direct this first question to you. What are the gold standard sources for defining PGX gene haplotypes?
Thank you, Christy. Yes. So there are a couple of consortiums that exist. One of them is PharmVar, which stands for Pharmacogene Variation Consortium, and CPIC is the other one, which is the Clinical Pharmacogenomic Implementation Consortium, which serve as the gold standard in the PGX references. This is due to their rigorous research and criteria. For genes that are not covered by these sources, additional sources can be recommended primarily by PharmVar, which are found on their resources tab on their website, and we also can curate that type of information. Other sources may also be useful in defining PGX gene haplotypes, but many of these will align themselves with PharmVar. The vast majority of resources will have used the allele definitions by PharmVar, but they may package them slightly differently.
Thank you, Anne. And Elias, let's come over to you for this next question. Does PGXI have any resources to help users query the PGXI downloaded content?
Yeah, absolutely. So the PGX download module really provides, either flat files. So typically those are TSV files that you can search the files manually. But even better, I think what the users might find beneficial is having access to a SQL file with database schema or MySQL schema. And using that, they can actually deploy the server locally or the MySQL server locally, and they will be able to run MySQL queries to query that data on their side.
Thank you, Elias. And Anne, I'd like to come back to you for this next one. Does PGXI support HLA genes?
So yeah, some of the viewers of this webinar and those interested in PGXI may know a little bit about HLA and the unique nature to these biomarkers. Currently, the PGXI content does not provide a full PGX details for all the HLA genes and biomarkers, but the PGX content does support partial SNP coverage for a subset of the HLA variants. In addition, if a lab is testing for at least one of the proxy SNPs that PGXI has in its knowledge base, such as a dbSNP ID that is associated with an HLA-specific star allele, then the genetic results with a clinical interpretation could be provided in the PGX reports. Additionally, if the lab is outsourcing genotyping of HLA genes and provides PGXI with the genotype used, then we can also support that type of content on the final report.
Thank you. And we have time for one more question. And this question has come in multiple times. So I'm going to go with this one. What file types are supported? Elias, would you mind answering this one?
Yeah, sure thing. So the main file types that are supported today are, of course, from NGS Technologies VCF files, and those could either be in genome builds 37 or 38. And we have built our KB in such a way that we have done the mapping. If you use 38 or 37, you'd not miss out on any kind of contact. So we have the leftovers performed as well. But of course, we do support microarrays or files from Array Technologies. And more specifically, the Agena Veridose. So the Agena Veridose software produces a file that combines data to produce star alleles as well as genotypes for genes that are not normally diplotyped. But we also have capabilities to support output files from Thermo Fisher. And as you might know, Thermo Fisher has two different file outputs, one that they call advanced genotyper and CNV files. Typically, these files are used in conjunction to understand individual variations as well as copy number variations of the CYP2D6 gene, but also the Thermo Fisher allele typer, which is an output of the allele typer software that calls star alleles directly, but also may include diplotypes as well as individual genotype calls. But here I'd also like to reiterate that even though we provide support for these files today, whether from arrays or VCF files, if you have any other file types that you would like to use on PGXI, we'd love to have that conversation with our services team and see how we can help you leverage our curated content on a day-to-day basis.
Elias, Anne, thank you so much for your time today. And did you have any final comments for our audience, Elias?
Yeah. So again, I'd invite everyone to visit the digitalinsights.qiagen.com /PGXI. Sorry about that. To have access or request a trial for the PGXI API. And then we'll be in contact with you where we can set up a multi-day trial so you can test out the API and see whether the curated content and insights that we have in our KB could help you reduce your turnaround time, and it will, of course, help you automate your PGX workflows.
Thank you again to Dr. Ann Bailey and Dr. Elias Hage for their time today and for sharing their expertise on how to rapidly and confidently translate complex pharmacogenomics data into evidence-backed insights to inform therapeutic outcomes. I would like to thank LabRoots and our sponsor QIAGEN for underwriting today's educational webcast. And before we go, I do want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Now, today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. To all our friends out there, thank you so much for joining us. Have a great day, everyone. Bye-bye.